Hello students, now we are going to discuss about the nutritional requirements for the plant tissue culture, right? So this is our new topics under the plant tissue culture unit 3 in pharmacognosy. So when we talk about the nutritional requirements, means nutritional is the sense nutrient which plant need for their normal growth and development, right? So whenever the plant, either it is grown into the in vitro or maybe in the open field and then somewhere, right, ultimately they need nutrient, they need food for their normal growth and development, for photosynthesis, for physiological action, for whatever the things they need to survive. So when we go for the plant tissue culture in the in vitro laboratories, right, in laboratory culture, what may be the requirements, what kind of things they need, that we are going to discuss and we are going to write in this particular topic, right? So let's start and try to understand what kind of things it is involved here, right? So nutritional requirement, it is very with the species, right? So the it's going to be the different, right? It's going to be the, if what kind of species we are going to cultivate, what kind of species we are going to have the culture, like it is a callus culture, single cell culture, root tip culture, shoot culture, what kind of things we are, what kind of species it is, opium or it is papaya, or if it is some kind of fruit, what kind of things we are going, according to that it is going to vary. What is going to vary? The nutritional requirement it is having specific for each and every species, even tissue from different part of the plant, right? So whatever it may be, root, stem, fruit, flower, bark, right? So in that case it is going to vary, may have different requirement. They will be having the specific requirements, some required macro elements, inorganic supplements, organic supplements, some vitamins, what kind of plant growth regulators, it is going to different, it is going to vary. So when we talk about the media composition, whatever the nutrient media we are going to prepare, what kind of composition, what kind of content, ingredient we are going to keep on that, that's important here to understand. The first one, it is the inorganic nutrient, then organic nutrient, then plant growth regulators, right? So coming to the first one, inorganic nutrients, we are having two types, macro and micro, right? So macronutrients we are having sodium, then phosphorus, potassium, calcium, magnesium and sulfur. So these things actually plant needs, so we are giving in the form of magnesium sulfate, zinc sulfate, potassium chloride, calcium chloride, right? Then ammonium nitrate. So like that things it is going to involve in that. So those things it will be subjected for whenever we are preparing the nutrient media for the plant tissue culture. Second is the micronutrient, as the name indicates, small, right? Macro means it is not that much small, right? So here we are having boron, copper, iron, manganese, zinc, molybdenum, right? So here you can see that ferric chloride, copper sulfate, molybdenum oxide. So these are the things which we are going to prepare, or while preparing the media we are adding these things. So from these things they are going to get, these are the nutrient which they are going to get, plants are going to get. Second is the organic nutrients. So when we talk about the organic nutrients, we are having again the series of things where we are going to understand, as the name indicate, they are organic in nature, right? So the first one, nitrogenous substances, when we talk about, so the series of vitamins we are going to add, like for example thiamine, like vitamin A, then pyridoxine, vitamin 6 it is called pyridoxine, then nicotinic acid, from there we can, the plant can get vitamin B3, calcium pentothenate, vitamin B5, then inositol, that also can be added in the media. Second, there are so many other things like complex nutritive mixture can be added into the nutritional nutrient media of the plant. Casein, hydrolysate, coconut milk, that we have seen into the history that the Vanderbilts, he has discovered that the plant can grow very good in the coconut milk and it can be useful for the media preparation. Corn milk, then malt extract, tomato juice, yeast extract, these are the things can be added, right? Then third one, in that organic nutrients we are having carbon source. So carbon source we can add the different types of carbohydrates like sucrose, glucose, fructose, then others also it is there, they like maltose, galactose, then mannose, then lactose, sorbitol. So these are the carbohydrate content which can be added for the carbon source. Activated charcoal also it can be useful, right? Then coming to the plant growth regulators we are having, right? So that also can be added in the media. That is also the nutritional requirements for the plant tissue culture. The first one that we have seen in the already plant growth regulator class. So auxin can be added like indole 3 acetic acid and indole 3 butyric acid, right? So many other nutrient it is also available that also can be added. For example, here in plant tissue in plant growth regulators we can have the gibberellins, it is there, right? GA3 it is generally recommended and it will be useful into the nutrient media. Then cytokinins G18 can be used. Then ethylene, this is the gaseous form which will be responsible for the ripening of the fruits, right? Then abscisic acid that inhibit the cell division, that also can be added sometimes whenever required, right? So these are the things can be added in the nutritional requirement. The plant need these things for their normal growth and development in vitro culture, so that can be added. Thank you all.